Mendel's work further inspired research into his findings. The fruit fly, Drosophila melanogaster, became an excellent tool for genetic studies. Similarly to Mendel's distinguishing characteristics such as height and color, scientists would study the inheritance of distinguishing mutations in Drosophila. Examples of this include curly wings, which were signified by a CY. This meant that the wings curled up and outwards. The ebony mutation gave rise to a black body and black wings in the Drosophila. Stubble, which was signified with an SB, was a mutation that caused thicker and shorter bristles on the back of the Drosophila. The white mutation, signified with a W, gave rise to a lack of pigment in the eyes, causing them to be white. The yellow mutation, signified by a Y, gave rise to a yellow body and yellow wings. Despite that Mendel's theories could be validated with pea plants, his theories were not applicable in Drosophila offspring, or other species for that matter. In an effort to solve this problem, a scientist by the name of Thomas Morgan experimented with Drosophila flies. He performed test crosses and observed the patterns of inheritance in the offspring. An example of one such cross would be the mating of a normal Drosophila fly with one that displayed the mutations for white eye, ebony, and curly wings. Let's say this cross gave rise to 1,000 progeny. The distribution of traits across this offspring cannot be explained by Mendel's theories. We see that the 9:3:3:1 ratio is not applicable here. Morgan was able to explain this through a phenomenon that is called genetic recombination. This phrase describes when chromosomes exchange portions of their sequences. This process most commonly occurs during meiosis, when corresponding chromosome pairs line up beside each other before being separated. The chromosome pairs form links, commonly referred to as chiasmata, and this allows for the exchange of genetic material. Hence, genetic recombination skews the figures in Mendellian inheritance. In order to stratify the risk of each particular recombination, according to the particular site at which the recombination occurred, Morgan studied the offspring of the test crosses that he performed to see what percentages of them displayed the characteristics of whatever particular recombination. Some were found to be more common than others. And so, let's say that this particular recombination has occurred in 20% of the progeny of this test cross. That means that this recombination has a frequency of 20% and hence has a 20% likelihood of happening during meiosis. So let's take a cell that falls within this 20% category. We see that it will have split into two cells with the according chromosome pairs. Hence, given that the chromosomes from each chromosome pair split into their own individual gametes, the percentages also split. This means that there is a 10% chance of achieving these particular gametes. Now let's look at the cells that fall within the 80% category, where the recombination has not occurred. And so, once again, the chromosome pairs split into their own individual cells, and when they split into gametes, the percentages further split. And so we see there's a 40% chance of achieving these gametes. The word parental is over each of these gametes because these are the gametes in which recombination has not occurred, and hence the gamete chromosomes are similar to those of the parents. We see that they each have a likelihood of 40%. And so let's observe what happens when these gametes fuse. Their likelihoods are multiplied to give an overall likelihood of 16%. The word recombinant applies to the gametes in which recombination has occurred, And so we see what happens when these gametes fuse. Their likelihoods are multiplied, and this gives 1% in this case, 1% in this case. And then, of course, let's observe what happens when a parental and a recombinant gamete fuse. 40% multiplied by 10% gives 1%. This demonstrates how Mendelian genetics is skewed by recombination.